So let's say we're a group of scientists and we all really love baseball, and we're really interested in this question of figuring out what is the nation's favorite team. And because this project is so interesting to us, we're going to try and figure this out by asking a bunch of people. But let's say that we're limited by the amount of money that we have and how far we could travel, and we only get to ask people in New York. So let's track their responses with this pie graph right here. And let's say that once we do this study, we figure out that we can break people's answers into, let's say, one of three different groups. So let's break this pie chart into about three different pieces. And let's say that when we do our study, we say that we have one really big group of people, we have a second group of people, and we have a much smaller group of people, which will give them the color green. So once we go through the data and we figure out that there's really three groups, the one in blue here, let's say that most people liked the Yankees, so I'll just label this Yankees. In the orange, we'll say that this group of people liked the Mets. And in this third group, we'll just put other, because there are lots of other teams that were represented, but the Yankees and the Mets were the two favorite. So since we did this study in New York City, and I'll just draw an arrow and label it NYC for being done in New York City, can we say that the nation's favorite team is the Yankees and their second favorite team is the Mets? And the answer is that no. We can't really call this the nation's favorite team being the Yankees because we only got these people to respond from New York City. And in fact, if we were to do a whole country basis, we might get something that looks more like this, with lots of other teams represented in varying proportions representing the many different teams that there are across the country. So this problem that we faced in our study, trying to draw a general conclusion using a group of people that were from a specific population and that weren't representative of the general population as a whole, gets a special name, and it's called selection bias. So I'll just write that in here, selection bias. And selection bias affects studies of all kinds, especially epidemiological ones. And in this video, we're going to talk about how selection bias affects some of the different studies we talked about in previous videos. But let's start with a more general definition of how selection bias works. So as we've discussed, all epidemiological studies get their power from comparing two similar groups of people. And in order for the results to be valid, not only do the groups have to be similar to each other, but they have to be similar to whatever population you're studying in order for there to have wide applicability of the results. So anything that changes the makeup of the group, either the controls or the experimental group, is going to give you some type of selection bias because your results won't be widely applicable to the general population since you can't draw a general conclusion. So let's go ahead and look at an example here. And let's say that we have two groups of people from which we want to select one side to be our cases and one side to be our controls. But if we just look at the makeup of the different groups, we see that we have a dissimilar makeup. They're not exactly equal to one another. So we have to do what we can as experimenters to make these groups equal to each other. So let's say we decide we want to get rid of this individual and these two over here on the left. And on the right, we're going to get rid of a couple or exclude them from being in our study. And all of a sudden, what do we have left but two equal groups of similar people? So now, since these groups are similar to each other, and I'll just put an arrow with the word similar, we can go ahead and do our study. And whatever conclusions we draw should be applicable to both groups because they have similar makeups in the cases and in the controls. If we had done our study with the original groups that weren't equal to each other, then our study would suffer from selection bias, because the way we selected our participants would have meant two different groups of people that we were comparing, and thus our answers would not be valid. Epidemiological studies can suffer from selection bias, and we're first going to look at selection bias in cohort studies and randomized control trials. So we're grouping randomized control trials and cohort studies together because they both have forward directionality. So let's just go ahead and write our timeline up here. And just as a brief review, we know they both have some kind of exposure in the case of a cohort study, or in a randomized control trial, you can call that a treatment that's assigned by the experimenter. And they both end up with some kind of outcome. And we're going to start on the left here. So I'll just draw our experimenter, starting with the exposure, and then following these individuals over time and tracking what kind of outcome develops. So how might we have selection bias in these two types of studies? So let's say that we start with these two groups of people, and let's say that the people in blue have the exposure or receive the treatment, so that's going to be our cases, and the yellow group of people does not receive that, so they'll be our control group. But over time, since we're following these people forward, some of them drop out of the study, or we can't reach them, so they're not responsive, or they're what's called lost to follow-up, which means you try and follow up with them, but you can't really find them because they've moved, or they've lost interest, or whatever reason. So let's say we'll just cross out, let's say we had some really bad luck, and 4 of these individuals dropped out. But at the same time, only 2 of the individuals down here did. So you might imagine that this is going to affect our outcome groups, because we're only going to have 1 blue individual left, but we're going to have 3 yellow individuals left. So then if we're at the end of our study, how can we compare 3 individuals versus 1? And this doesn't really make sense because our answers are going to be all sorts of skewed because we don't have an equal number of people or equal groups in each of our outcome populations. So because they're not equal, and I'll just write here, not equal with an exclamation point, this is an example of selection bias because the composition of our groups has been changed by some things that were out of our control. At the same time, selection bias also affects case control studies. So let's just go ahead and get case control on the top of this page, since that's the type of study we're going to be dealing with. And like we did with cohort studies and randomized control trials, we'll just go ahead and draw our timeline. And on the left-hand side here, we'll mark our exposure that we're interested in. And on the right-hand side, we'll mark our outcome. And just as a quick review, we know these have backwards directionality. So the case control study starts after the outcome is known, and we're looking back to see what type of exposure caused that particular outcome. So in this example, let's say that we're really interested in the liver disease cirrhosis. So I'll say this purple individual has cirrhosis. So I'll just mark him down as having cirrhosis, and we're going to compare him against this individual who does not have cirrhosis. So I'll just write cirrhosis with a line through it. And we're really interested in how cirrhosis relates to a previous exposure with a certain amount of alcohol. So let's just say this bottle here represents alcohol, and this no means that no alcohol exposure or reduced alcohol exposure was noted for this individual. So like we've talked about before, we're going to see how many people in the cirrhosis group didn't have exposure, and the same thing for the non-cirrhosis group. And we're going to see how many in the cirrhosis group had alcohol exposure, and how many in the non-cirrhosis group also had alcohol exposure. And by comparing how many of each group had or didn't have alcohol exposure, we can get some idea of what kind of a risk factor alcohol exposure is. But let's throw a little wrench into this and say that because of whatever reason, we decide that we can only conduct this study at a hospital. So that's just what this white box means. So let's say this is the hospital, and we'll just color in our red cross here to symbolize hospital. So this particular study may suffer from selection bias because it's possible that if you're in the hospital, although you're not suffering from cirrhosis, you may have a higher baseline alcohol exposure than other people in the general population. And that may or may not contribute to why you're in the hospital, but the point is that your alcohol exposure is different from the general population, so you can't really say that whatever results you come up with in this study are widely applicable. You can really only say that for this group of patients that are in the hospital currently, alcohol is or is not a risk factor for cirrhosis or not, depending on what you find. So again, like we've been talking about, for selection bias, anything that changes the composition of the group or makes it different from the group to which you want to apply an answer is going to skew your results and give you selection bias.